Yeah. Hi, everyone. Thank you very much for joining our webinar. My name is Olga, and I will be hosting this webinar for you. And before we start, I will do a few announcements. Yeah, first of all attendees will be muted during the webinar. And if you have some questions or comments, you can type them in the chat box or questionnaire box, and once the talk is completed, I will read your questions and the presenter will answer them. Yeah, if there are people who can't join us today, the webinar will be recorded and everyone can get access to the record on our YouTube channel. Yeah, please subscribe to our YouTube channel and do not miss the great content about drug discovery. Just a few words about ChemSpace, who we are and what we are doing. So ChemSpace, it's a one-stop shop for small molecules, where the researchers can fulfill all their needs in small molecules. And our main goal is to simplify the purchasing process for our customers when they can place just a single call and get compounds, small molecules from different suppliers all over the world. Our catalog includes over 400,000 of building blocks and reagents, over 6 million in-stock compounds, including 20,000 tool compounds. Besides in-stock compounds, we offer billions of make-on-demand analogs to in-stock compounds. These make-on-demand analogs can be synthesized by our valuable partners, chemical providers, and the synthesis can be performed with a rather acceptable period of time. To search within these billions of molecules, we implemented a really fast and efficient search engine on our platform. You can use different type of search. You can do similarity search, substructure search, text search. Different identifiers are also possible. You can import the file and get the results within a few minutes. Recently, we enabled search of tool compounds by trade name. And it's very easy. You just need to type, let's say, trade name Glycate and press the button search. And within a few seconds, you will get the results. You can see that this compound is offered by a few suppliers. You can scroll, you can filter, use different filters on the left, and you can also like choose the price. You can choose the package size, and pressing one button, you can add these compounds to the cart and proceed with purchasing. If you still do not find the right molecules, or you're having some troubles upon this search, you can always contact our account managers. Our team is very experienced in sourcing compounds because we have well-established sourcing and procurement workflow. We can source single molecules and we can source a list of compounds. More than that, we are always happy to assist with compound selection. So you can get the offer with a small number of suppliers, but still with a good coverage and the best price. We assist also with formatting, consolidation, and customs clearance. There are a few special sourcing programs we offer to our customers, like providing building blocks for library synthesis in small micromole amounts. We also provide screening compounds for virtual screening projects with a really fast turnaround time. But there are always some requests for not small molecules, but for something else. And due to our partners in different areas, we can fulfill different customs requests, like large-scale synthesis, DNA-encoded libraries. We can offer synthesis peptides, products, antibody-drug conjugates, microsite, and also glycosylated small molecules. And I'm very happy and I'm honored to introduce to you our guest speaker, Jürgen Hansen, who is Chief Scientific Officer and Chief Executive Officer of Riverstone Biotech. He has extensive experience working in different biotechs. For 5 years, he ran like 5-site CRO with main focus on molecular biology. And today, he will be giving a talk about improving characteristics of small molecules by enzymatic glycosylation. With that, I will stop my presentation and give a word to Jürgen.
Okay, thank you very much. It's a pleasure to have the opportunity to tell about something I have worked on and off on for, I was calculating today, about 18 years of my life, namely glycosylation of small molecules. Originally, I am a molecular biologist. I have a PhD in genetics from the Carlsberg laboratories. But since 2003, I've actually been working in various biotech companies on making small molecules by synthetic biology, but also on improving small molecules by different means, and in particular by glycosylation. So it's not really moving. Now, so small molecules, I guess you all know, do not always behave like we want them to. And there are different ways to improve on that, but one is glycosylation. And let me start by, on this slide, to tell you a little bit about how you can improve small molecules by glycosylation. Maybe some of you know all of this, but then bear with me. So if you start on the left bottom corner, you can, most well known, increase solubility of hydrophobic molecules by attaching carbohydrate molecules to them. The more the better. If you go up to the left side, it is also known that you can improve on stability of certain small molecules, both when it comes to chemical and biological instability. This is an example with a peptide that had a carbohydrate sitting at each end. If you go up one more step, you can also imagine, and in some cases it's been shown, that you can do tissue targeting. And that is because certain carbohydrates are the epitopes recognized by human receptors and animal receptors in general. If you go to the right on the top, you can get an altered binding on mammalian receptors, and by altered binding, of course, you get a changed functionality. I would say that a really well-known example of this is, well, I work with this, that's why I know about it, but maybe you heard about stevia sweeteners, which is a natural sweetener from a plant called sweet leaf. The left molecule is the A-glycan, so the one without any carbohydrates sitting on it. It's called steviol. It does absolutely nothing to the human taste buds. If you add 2 glucose molecules, as in the middle graphic, you get a quite, I would say, very sweet molecule. It's called stevioside. And if you keep on adding sugar molecules, just one more makes it into a riboside that is a lot more sweet. Then if you go down, you can in some cases get better bioavailability that has not just to do with improved solubility. This is an example where you, by adding a sugar molecule to a drug, can get better penetration over the blood-brain barrier. and that's because there are transporters, importers, exporters that recognize carbohydrate motifs. And the final example to the right bottom is that you can add a sugar to a drug molecule and thereby make a prodrug that will give you controlled release of the drug in the body, because the bond between the carbohydrate and the drug gets cleaved by enzymes or certain chemical conditions. So in accordance with that, many existing molecular drugs require sugar units for their effects. These are just 4 examples. There's the cardiac drug digitoxin from digitalis. This is a triterpene with 3 sugar molecules attached. There was a very, very nice paper coming out some years ago where researchers tried to take these 3 molecules away, these 3 carbohydrate molecules away, and put others on. But only in these particular 3 sugars in this particular configuration gave the particular cardiac effect of digitoxin. To the right, you see clarithromycin, a well-known antibiotic of bacterial origin. Also, in this case, the effect is linked to the molecule with the 2 sugars sitting on it. The left bottom, you have heparin used as an antithrombotic, and basically that is a chain of carbohydrates. And finally, you have etoposide to the right, which is a semi-synthetic. It's a natural molecule that's been changed a little bit, but part of, or a very big part of the functionality is the, I would call it semi-natural sugar sitting on the top. Then there are many different sugars, of course, in this world, and I put a few of them out to the right: glucose, galactose, glucuronic acid, xylose, N-acetylglucosamine, rhamnose, and several others. They each can confer different things to small molecules. For example, glucose, mannose, and glucuronic acid may all increase the solubility of a small molecule or help in transport and change the activities of therapeutics. Sialic acids, galactose, xylose, and N-acetylglucosamine are sugars that are really well known by receptors in mammalian bodies. So these are the ones you go to if you want to start messing with tissue targeting. Muramic acid is an interesting carbohydrate found in bacterial cell walls, and that is why it and related molecules confer a strong immune response in mammalian and human bodies. So you can think of that molecule if you want to do therapeutics with immune modulating activity. Amino sugars such as monogerymycin, you can see it on the right low, is in consideration as antivirals, but also as immunomodulators. So that's also an interesting sugar to think of using. And finally, deoxyhexoses are used in decoration of antibiotics, flavonoids, etc. in nature. So I will come with a bold statement, and it's often provocative. If you have an audience of chemists, I will nonetheless go there and say that in my and our mind, enzymes provide the preferred way to glycosylate a small molecule. And I'll try to convince you that that is the case. So chemical glycosylation often involves quite tedious protection and deprotection cycles, because if you have more than one glycosylable site group, you need to protect the one you don't want, glycosylate, and so on. It's a difficult approach, it's expensive and pretty slow, and therefore not viable for many intended uses. Enzymatic glycosylation, on the other hand, circumvents such challenges. An enzyme that adds a specific sugar to make the desired change can almost always be found in nature. Enzymes, we think, give a selectivity that chemistry can rarely match. Even stereoselective enzymes can be identified. I've worked with such enzymes that will only glycosylate and do that to 100%, one of 2 stereoisomers of a small small terpene. Enzymes can put sugar onto sugar that's sitting on a small molecule, and they can do that with precise bond specificity, so 1,3, 1,2, 1,4, and so on. It offers what we think is a greener production, because you are just messing with enzymes in watery solutions, so there's no need for harsh chemicals or reactions, and that's why we think that enzymes for glycosylation is will normally be much cheaper than doing chemical glycylation. So a few examples of improvement done on small molecules by glycosylation. These are all from the literature. So solubility, the first example. So curcumin is, as you probably know, it's the yellow agent of turmeric. It's basically a brick. It has virtually no solubility in water, or very, very low. By just adding one glucose unit to one end of that molecule, you improve solubility by 230 times. If you add 6, 3 on one side, 3 on the other side, sitting in a chain, you get a 22 million-fold higher watery solubility. An example in what we call drug delivery, vitamin E usually does not diffuse well through skin, and that is desired in certain applications. If you glycosylate that, just put one glucose on vitamin E, it does diffuse through skin and is then metabolized to the active form once inside the cells. Blood-brain barrier penetration. There was a nice study where it was shown that endorphins, these small peptides, are much better taken up over the BBB, and therefore the glycoside gave better analgesic effects. Dexamethasone is used when you have certain inflammatory conditions in your bowel, and often or almost always you have to take a lot of it, because only a little bit of it reaches the lower bowel, about 1% in most cases. If you glucosylate dexamethasone, on the other hand, at least in one case, it's shown that 60% reaches the bowel, and here it's cleaved to the active form by enzymes made by the resident bacteria. Finally, it's interesting that morphine itself is actually not the most active form of this alkaloid once inside the human body. It's rather the morphine glucuronic made by enzymes from ourselves that is active. It's 45 to 60 times more potent analgesic than morphine itself, and with a longer lasting effect. And at least some authors think that really that's the form that does something in terms of pain management. And this page shows the references to the papers for the findings I just went through, plus a lot of others. So let's talk about nature's glycosylation machinery. So efficient and specific glycosylation is very much part of nature's fabric, much more than most people realize. All cell walls in all living cells and most natural storage foods are made by glycosylation to form particular polysaccharides. And two examples of cell wall components are obviously cellulose, but also chitin, which forms the skeleton of, for example, crab shields and insect exoskeletons. And then obviously starch and glycogen as storage polysaccharides in animals and other organisms. Many others than these exist. In addition, there's a vast number of proteins in nature that rely on glycosylation for both stability, functionality, and targeting. Examples are antibodies, major histocompatibility complex, platelet proteins, but there are many, many others. Finally, small molecules are glycosylated for storage, for transport, for functionality, or detoxification disposal. And I have here 4 examples of this. animals, such as ourselves, glucuronidate synbiotics for clearance in our kidneys, insects, glycosylate odorants for fast elimination from the olfactory tissue, and that's actually strong evidence that the same happens in mammals. Bacteria and fungi use glycosylation to fabricate potent antibiotics. I mentioned that a few slides ago. And finally, plants glycosylate to detoxify symbiotics. Imagine being a plant. You cannot run away, so you need to have ways to avoid chemical warfare on the ground. And that's why they have such efficient machineries to detoxify, to secure defense compounds for the same reason, and to optimize functionality of secondary metabolites. I put a green bracket or green square around these 4 examples, and that's because all of this is done by a certain class of enzymes that are called family 1 UDP-glucose glycosyltransferases or family 1 UDTs. And next I will talk a little bit more about those. So family 1 glycosyltransferases, UDTs, they turn small molecules into a number of different glycosides, you could call them. They do O-glycosides if there is a hydroxy side groups on a small molecule. They can do sugar esters if there is a carboxy group sticking out. They can make glycosamines if there are appropriate amino groups sticking out, and they can make thioglycosides if there are appropriate thiol side groups sticking out. They can even, in some cases, do what you could call permanent glycosylation, so C-glycosylation, a sugar sitting directly on a carbon, which will not fall out or fall away or be clipped away by any enzymes. And the prime example of this is carmine, the red colorant used in so many things, among those lipstick and Aperol for Aperol spritz. It's a very interesting reaction, and there's a lot of research undergoing in that. These enzymes do all this by using what we call activated nucleotide sugars. For example, UDP-glucose, and UDP stands for uridine diphosphate. You can see the molecule to the right in the green bracket. A UDP-glucose a UDP enzymes takes the sugar sitting on a UDP-glucose and puts it on to a side group of the substrate, and thereby releases UDP and the glycoside. UDTs, the whole family 1 UDTs are what we call inverting enzymes. So whereas the glucose in UDP-glucose sits in an alpha configuration to the rest of the molecule, in the resulting glycoside you have a beta configuration because you have this inversion going on. Animal UDTs tend to work on many substrates. That's also why you see that animals such as ourselves, we don't have many UDTs, we have a small handful, whereas most plant UDTs are quite specific, even to the point, as I said, where only a specific stereoisomers of a small molecule get glycosylated, and that's why they have a lot of UDTs. So any given plant species on this earth have between 80 and 200 particular UDT enzymes. With perhaps 400,000 plant species available, the enzymatic diversity is simply enormous. So let's talk a little bit about the substrate structure diversity of plant UDTs. So depending on the sequence homology, plant UDTs are divided into subfamilies. We named them 71 to 94, and there are even a few further now because this was not enough in the end. And these are again divided into subfamilies and further on. These divisions, to I would say a limited extent, reflect substrate specificity. So in a few subfamilies, you see that these enzymes are most often good at certain chemical groups, such as polyphenols, carboxylic acids, lipids, or other glycosides. So doing di-glycosylation, putting sugar onto sugar. But most often you cannot really predict what happens. But from our experience, and as I said, that's about 18-year-old, and the literature, it seems that most structures with appropriate site groups can be glycosylated. There are limitations though. We have never seen amino acids being glycosylated, and that's probably because they are sort of sacred or holy compounds in living cells. If they were glycosylated to any large extent, it would mess with the protein synthesis machinery. And also UDTs in general do not work with very large molecules, very large small molecules. So we've never seen anything more than a molecular weight of about 1500 being glycosylated. But we have seen many molecules in the around 1000 to 1200 molecular weight being glycosylated. We do not see any systematic difference between synthetic and natural small molecules. It seems to be they seem to be indifferent about this. One thing to keep in mind though is that if the folding of the small molecules in a given solution is so that the only available site group is sort of shielded from the environment, then things get difficult. There are things to do about this, but it does get difficult. Some examples from nature below on the bottom of this slide. Vanillin glucoside. This is actually the form of vanillin that you find in the vanilla part before it gets fermented and dried and so on. Doxorubicin, the cancer compound. Crocin, which is really a saffron color, has 4 glucose units sitting on it. Pasicocin is so-called cyanogenic glucoside from a plant in nature. It is a defense storage compound. If the glucose gets cleaved away, the A-glycan deteriorates to give out cyanohydrin. And finally, an example of a drug glucuronide, as it will look after a trip through our body. So let's talk a little bit about what our company, Riverstone or Glycate, can offer. So we have made, and again, this relies on a number of years of research. We've made a UDT library with a large number of different enzymes. As I alluded to, it is really not easy to predict which enzymes will glycosylate which small molecule. So you really need to screen a large panel of enzymes to give yourself the best chance of success. The bulk of the enzymes we have collected, which is no less than 402 UDT enzymes, were selected from public sequence data based on 2 criteria. We wanted to have as much taxonomic diversity of the source organism as possible covered, and that is from the perspective that if you cover many different levels of taxonomy, you also cover many different habitats and niches, and thereby a lot of different biochemistry. Second criterion was to represent all known UDT subfamilies and subfamilies. So using these 2 criteria, we found the first 402 UDT sequences. In addition to these, we took 48 UDTs that we have history with, where we already knew a bit about their activity. So some of these are what we call household UDTs that do work on quite a lot of substrates. Some were very specific UDTs that we knew worked with specific, you could call them exotic substrate groups. Mostly these enzymes came from Arabidopsis and stevia, the sweet leaf I talked about before. The combined library, we see a probability of glycosylating any substrate molecule of about 75%. Again, that's within the limitations I mentioned, for example, a molecular weight below 1500. And below you see pictures of some of the sources these enzymes came from. So we call this the Glycate. It contains 450 enzymes in total. We have 4 96 well plates containing 380 of these enzymes, of course with 1 unique UDT enzyme per well, plus a positive control molecule for validation that nothing bad happened to the collection. Then we have 1 additional 96 well plate with 70 enzymes that are particularly likely to do di-glycosylation, so adding a sugar to a sugar already sitting on a small molecule. You can run assays like this in many different formats, and I can tell that 18 years ago I personally did it using TLC plates with radioactive sugar donors and X-ray films after that. We've come beyond that, and so we use and we recommend using 2 different ways. One is to use a plate reader that gives you simply a measure of how much UDP is released through the glycosylation reaction. It's fast and easy, but you also only get qualitative information, but it's good for a fast screening. Better, I would say, is to do HPLC or LCMS or whatever you need to use of more advanced analytical instruments that gives you quantitative information on your hits. But of course, it's a little bit more resource heavy and slower. In any case, what you do is to take the small molecule substrate, add it with 1 enzyme, the sugar donor, incubate for 4 to 30 hours, and do your analysis. These are just a few results from our screening. We have many more, of course. So this shows how many out of the 380 original enzymes we collected gave hits with different molecules. So in the middle top, you can see that with trichlorophenol, which is one of our what we call positive control molecules because it works well with so many UDTs, we get about 158 actives. With etoposide to the left of that, we got 11 actives. With quercetin, flavonoid, to the right, we got 64 actives. And then with triptolide, 1, valeric acid, 3, and the alkaloid oripavin, 1 active. So what do you do when you have a hit? And I should say the things I go through now are things, some of the things we can do with our partners, some of them we cannot, but in those cases we can advise. So when you have a hit, you want to establish which substrate site groups have been glycosylated, at least in the cases where you have more possible glycolysable site groups. You do that by making a little bit of the glycoside and purifying it and doing NMR. We can help with that. Make more of the glycosylated molecule, yes, for that purpose, but also if you want to test the glycosylated molecule for improved functionality, so altered solubility, altered, what do I know, colors, and so on. We can also talk about adding additional or different kind of sugars than just UDP-glucose, which will usually be the first you test with. Then we work with a British group of computational chemists who are very good at working at doing computational guided molecular biology, in particular with UDTs. And this is something we turn to when we have a hit enzyme that, however, is not very good with the substrate molecules, and we want to make it better or maybe more specific, and so on. We can help with that. We can also, to a limited extent, help with assessing IP and regulatory opportunities. What we cannot do ourselves is to scale up glycoside production beyond, let's say, 100 to 200 milligrams of glycoside. If you want to make grams or multi-kilos or even tons, you have to go to different people. But we have ideas and contacts. microbial transformation, where you express these enzymes inside microbes and do a biotransformation. That's something we do for a living, and we can help with that. And in vitro transformation is basically a scaled up version of what we did in the screening. So I think we missed this one, sorry. The screen is available as a product or a service. This should have been before the last slide, I'm sorry about that. So we can provide the kit as a product with support, and that support can be online or through phone. But we can provide the kit so that you can run the assay. The kit pricing depends on its size, which again means how many compounds can you test, and the price range is between 40,000 and 75,000 euros. We also do service options, so you can ship the compound to us and we can run the assay as a service. The screening service pricing depends on the complexity of the molecule, and by that I basically mean solubility and toxicity, but also the related analysis. So is it simple HPLC, is it complex HPLC or LCMS? So the range for doing such a service is 18,000 to 25,000 euro per substrate molecule. In all cases, we will advise how best to use the kit for your particular program goal. We will also advise against a particular substrate if we think it is really not very likely that it will work. Given you wish to progress after positive results, we can supply you with the plasmids for that can express the UDT enzyme in question. That's for a modest fee. We can also make more enzyme for you for a price. Most importantly, perhaps, we make no downstream claims for any further services or fees, and we assert no IP claims. You own any potential composition of matter or use claims. Of course, you have to look into what's out there. Just maybe by chance, someone in the meanwhile filed a patent on one of these enzymes. I would find it unlikely though, but certainly we do not assert any claims. And then we should have had this one, but we've been through this. So thank you very much for listening to this talk. It was a pleasure. Here are our contact details, and you can go to www.gly-it.com, glyate.com, to see a bit more. There you will also find the references for interesting literature that I went over. Thank you.
Thank you very much for really interesting talk. I enjoyed personally, and I hope all attendees did the same. If anybody has any questions, please type them in the question box. And I actually have a small question. So you mentioned that amino acids, it's a limitation for your method. Yeah, I understand this. But if we are dealing with like molecule with a lot of functional groups in it, is it okay? Is there any limitation using your kit?
Is there a limitation if you have many glycolysable groups?
Yeah, if we have many like functional group in the molecules, like hydroxy group, amino group, and something like that.
So what we have seen in the past is that when you test enough enzymes, you will almost always find one enzyme that glycosylates one side group, another enzyme that glycosylates another side group. And we have certainly seen with small molecules that contains both an amino and a hydroxy group that we have enzymes that do either of those, but usually not both. If we have many of the same type of groups, so many hydroxy groups, which is most often the case, we see enzymes that will glycosylate both, and we see enzymes that will glycosylate one or the other. And if you have small molecules that have a carboxy group and a hydroxy group, we will almost never have one enzyme that will do both, but we will usually find one enzyme that does one and one the other.
Yeah, thank you very much. Thank you very much for your answer. And another question from me. So your kit contains like a certain number of enzymes. Is it correct? Yeah,
That's
and
right.
you said that we can get like a different, absolutely different number of hits. Is there any cases when you don't get any hits at all?
Yes, there are cases when you don't get any hits at all. So that's why we say that with this library, so far, we can see there is a 75% probability of getting a hit with any given small molecule we put into the screen. So there are 25% of cases where we do not get a hit. But those are most often substrates where we were already a little bit worried about the side group sitting protected by the folds of the molecule or something similar. I would say that in the cases where something looks like it's available, we can usually always get a hit.
Okay, with one kit we can test like one small molecule, is it correct?
With one screen you can test one small molecule, and then if we sell a kit, it will be for I think it is for either 10 reactions or 40 reactions. So with a small kit you can test 10 molecules, with a large kit you can test 40.
Mm-hmm. Yeah, but it seems that it's much cheaper to actually contact you and send the small molecules to you and allow you to run the screen.
It depends very much how comfortable you are, a given group of people are with this kind of technology. I mean, it's not. I wouldn't say this is complex in any way, but if you're not used to doing stuff like this, it is probably both. It's certainly easier, and it's probably also cheaper to let us do it. Yes.
Yeah, and another question about application area. I mean, so I believe that once the company who is working in drug discovery has the lead compound, they come to you with certain problems, let's say solubility or something else, and you help to improve. Is it correct?
Yes, that's the idea. Mm-hmm. But I would say normally we do not advise on what happens to a small molecule once it gets glycosylated. We just say that there are a number of possibilities, and what we can help with is to glycosylate it so that you can test it.
Yeah, and last question from me about medium. So, you know, do we work in like water or some buffer solution, or you can use like DIMSO or something like that? What about solutions for the reaction?
So the glycosylation reaction?
Yes.
So you can go up to 20% DMSO. Which is why it is actually possible to work with rather insoluble compounds. So fortunately, these enzymes are very robust, at least when it comes to DMSO. So that's our go-to. But we also have used in the past certain surfactants that will also help you in presenting your substrate to the enzymes. But it is a watery solution with up to 20% DMSO or something similar that you need to use.
Okay, yeah, thank you very much.
You're welcome.
Another question, how do you make predictions for enzymes? Will it work with defined molecule or not? Is it empirical or in silico?
So it is mostly empirical, and that is why we have chosen to collect such a large amount of enzymes. I will talk about one exception. If you have let's say you have a flavonoid with a glucose or a rhamnose sitting on it, you often find those in nature, and you want to see what happens if you add another glucose to that xylose. Then we do have some predictability we know the subset of enzymes that you would need to use for that kind of reaction. And that means that to increase your chances, it's possible to source more enzymes of that kind. There are also some predictability, as I said, when it comes to certain lipids and to certain other molecules, such as flavonoids. But outside that, with a synthetic molecule, for example, we have no predictability.
Mm-hmm. Yeah, I see.
It's empirical.
Yeah,
and that's the strength of our platform, that it allows you to get success with an empirical method.
Yeah, it's true. Okay, the next questions. What is the difference in glycosylation with glucose and other sugars? Actually, I believe that you mentioned it over your presentation. Yeah.
If you want to start, I mean, we're actually working right now with a partner who are interested in turning certain molecules into something that's relevant for the human body, and there you can say you would want to work with certain sugars rather than other sugars, and sialic acid is one good example of that. Because it is part of many signaling processes in the human body. N-acetylglucosamine is another example, because that particular sugar is sitting on many proteins in our body. So you can do a little bit there. Whereas if you are mainly after just improving solubility, you would instinctively go for glucose, also because that's by far the easiest sugar to work with. The sugar donor is least expensive, and you have the highest success rate with glucose. So certainly there is some knowledge about this, but it is very much experimental.
Okay, thank you very much. I don't see any other questions. Yeah, so it's very interesting to listen to you, and yeah, thank you very much. If anybody might have questions afterwards, you can contact us over emails. You can see right now on your screen, and as I said, the video will be available like tomorrow on our YouTube channel. So thank you very much, Jürgen.
Thanks very much for the attention and possibility.
Yeah. Bye-bye.
Goodbye.